Hello dear students, today we are going to learn about fertilization. Fertilization is a process of fusion of male and female gametes to produce a zygote with diploid number of chromosomes. Male and female gametes have haploid number of chromosomes. Male gamete has either 23 plus X or 23 plus Y number of chromosomes. On the other hand, female gametes has 23 plus X chromosome set. After fusion, they produce a zygote with 46 plus XY or 46 plus XX set of chromosomes. Fertilization takes place in the ampulla of the uterine tube. Ampulla is the broadest part of the fallopian tube. This is the ovary. These are the fimbriae, and this is the uterine tube, and this is the broadest part, ampulla. Fertilization has three phases. First is the penetration of corona radiata. Second is the penetration of zona pellucida. Third is the fusion of oocyte and sperm cell membranes. This is the secondary oocyte or ovum, and this is the zona pellucida which surrounds the secondary oocyte. Zona pellucida is a glycoprotein, thick glycoprotein cover, and this thick glycoprotein is surrounded by cumulus cells which derived from the granulosa cells of the ovary, and these cumulus cells arrange in a radiating manner surrounding the ovum, and this is known as corona radiata. Before fertilization to occur, priming of sperm is necessary, and it consists of capacitation and acrosomal reaction. So the first step in the fertilization is the capacitation of the sperm, followed by penetration of the corona radiata. Capacitation is the prerequisite of fertilization. So only capacitized sperm can penetrate through the corona radiata and produce acrosomal reaction. It takes around 6 to 7 hours to complete. So there is no benefit to come first in the race of sperms. Only capacitized sperms which has passed 6 to 7 hours in the female genital tract are able to penetrate the corona radiata, and this capacitation takes place in the female genital tract, mainly in the uterine tube. And mechanism of capacitation includes the efflux of cholesterol from the plasma membrane of the sperm head, calcium influx, bicarbonate influx. It causes the removal of glycoprotein cover from the surface of the sperm and removal of seminal plasma proteins from the sperm. And in this diagram we can see the first step is the removal of decapacitization factors, mainly the glycoproteins, then removal of cholesterol from the plasma membrane of the sperm. Then due to this, increased membrane fluidity occurs and raft aggregation occurs with enhanced zona affinity. Affinity of the sperm for the zona pellucida increases, and after this docking of the acrosome to the zona can takes place. On the other hand, tail gain increased mobility with the activation of tyrosine kinase. Second step is the acrosomal reaction, with the penetration of zona pellucida. Sperm bind with the zona pellucida with the help of ZP3 protein. This is ZP3 protein which is on the zona pellucida, and sperm head has the ligand for this protein. So the sperm entry takes place only with the attachment of the zona pellucida to the sperm head. After this, there is release of hyaluronidase and acrosin occurs, and due to which penetration of zona pellucida occurs, and his sperm head comes in contact with the oocyte membrane, and then the release of lysosomal enzymes from the cortical granules lining the plasma membrane of oocyte occurs. This is known as zona reaction, and it changes the permeability of zona pellucida to prevent further sperm entry. Here in this diagram we can see this is the sperm head which has this blue dot which is known as acrosomal granule, and this is the green part is the nucleus, and this is the tail, and here is the mitochondria of the sperm. Now this sperm, when attaches to the zona pellucida, after attachment it releases the acrosomal enzymes. These acrosomal enzymes consist of acrosin, hyaluronidase, and with the help of this, penetration of zona pellucida occurs. After the penetration, when sperm reaches the plasma membrane of the oocyte, the granules of the oocyte releases content which is the lysosomal enzymes, and this content changes the nature of the zona pellucida and make it impermeable to further sperm entry. So here we can see these cortical granule release is necessary for the zona reaction, and this cortical granule release only when a sperm enters and reaches the oocyte membrane. After this, the fusion of oocyte, the third step is the fusion of oocyte with the sperm cell membrane. Fusion of oocyte occurs with the attachment of the integrin of ova with the disintegrin of the sperm. Sperm head and tail enters in the cytoplasm of the oocyte, and plasma membrane left behind. Reaction following the sperm entry in the oocyte includes the cortical and zona reaction to prevent polyspermy, that is the release of cortical granules, and zona reaction which prevent the polyspermy. No further sperm can enter now, and then the second step is the second reaction is the oocyte completes second meiotic division, that forms the definitive oocyte from the secondary oocyte with the female pronucleus and a second polar body form. Third reaction is the metabolic activation of egg. Fourth is the oocyte and sperm nuclei swell to form pronuclei, and fifth is the pronuclei membrane dissolve and nuclear fusion occur to produce a diploid zygote. Thank you for listening, dear students, and these are my references.